Well, good morning, everyone, and welcome to Frontiers in Oncology. Thank you for joining us. It looks like people are just getting on the meeting now. Of course, it's an unusual day, being election day, but it's our great pleasure to be hosting Dr. Steven Rosenberg from the National Cancer Institute. I was just chatting with Dr. Rosenberg and learning a little bit, having some very few insights into his early career, which is fascinating. And let me share some of those with you. So Dr. Rosenberg was born in New York City, went to college at Johns Hopkins and then to medical school at Johns Hopkins. He then did a surgery residency at the Brigham Hospital in Boston. And when he was done with that, he did a PhD in biophysics, where he studied erythrocyte proteins. And I was just asking him if this was a common phenomenon for surgeons to be doing PhDs. And of course, back in those days, it was not. This is all pre-MSTP training time. And Dr. Rosenberg left his surgery life behind during this period and focused intensely on his PhD studies. And then he transitioned to the NCI, and after extensive training in surgery, his first job was the director of surgery at the NCI, a job that he never left. And so he was joking that this was his first and his last job. So really a remarkable career. This was in 1974. Of course, Dr. Rosenberg has pioneered immunotherapy for cancer, and he has many, many awards, too numerous to count, but one I'll call out, which is the American Cancer Society Medal of Honor, which he received in 2015. Dr. Rosenberg has done incredible work in adoptive transfer of tumor-infiltrating lymphocytes into patients to treat patients with advanced cancer. And I remember reading his papers from years ago and seeing the remarkable results that he got in patients with melanoma. And this was at a time long before checkpoint inhibitors were being developed. And it really convinced me that melanoma was a disease that was responsive to the immune system, seeing his beautiful data. So it's a pleasure to welcome Dr. Rosenberg today, and we're looking forward to his talk.
Well, thank you. I'm going to discuss the development of cellular immunotherapies for patients with cancer. As you've just heard, I've been a government employee at the NCI for my entire career, so sadly I have no personal disclosures. The goal that I'll discuss is the development of effective immunotherapies for patients with metastatic cancer based on the adoptive transfer of immune cells with anti-cancer activity. And it's this cell transfer therapy that we've emphasized over the years, and it has a variety of advantages over other treatments, non-specific stimulation of the immune system with IL-2, inhibition of checkpoint modulators, cancer vaccines, another approach not yet fully developed. But this adoptive cell transfer therapy has a variety of advantages. One, you can grow very large numbers of highly selected cells in the laboratory with a very high avidity for tumor antigens. Because you have the cells in a test tube that you're administering to patients, you could potentially identify the exact cell subpopulations, effector functions, antigens recognized that are required for cancer regression in vivo. And finally, and perhaps most importantly, you can manipulate the host prior to cell transfer to provide an altered microenvironment for the transferred cells, because the reagent that's being used, this living drug, the patient's own cells with anti-tumor activity, have been removed from the body, and one is then able to treat the patient, in our case, with a lymphodepleting chemotherapy that turns out to be essential for seeing durable, complete responses in patients with cancer receiving this kind of cell therapy. Now, I know in this audience, I don't need to emphasize the difference between conventional T cell receptors, which are alpha and beta chains on a T cell that recognize a processed peptide on a surface MHC molecule. Now, the first cell therapy to be approved by the FDA, however, utilized something called a chimeric antigen receptor, And again, we look at the variable regions of the heavy and light chain, which are turned into a single chain. This was a technology originally developed by Zella Geshar and Gideon Gross at the Weizmann Institute. That single chain, which now conveys recognition based on antibody recognition, is then attached to intracellular signaling domains and can actually convert the T lymphocyte into a cell that can recognize antigens based on this single chain antibody. And I'll spend just a moment on this to make a point about the value of CAR T cells potentially in epithelial cancers, and present therefore this first patient ever to be successfully treated with a CAR T cell here at the NIH. This was a 48-year-old male with a non-Hodgkin lymphoma. He had undergone multiple chemotherapy regimens, a SPACE regimen, a vaccine, checkpoint modulator, an EPOCH-R combination chemotherapy, and had progressed, came to the NCI for treatment with his autologous anti-CD19 CAR T cells. These are CAR T cells utilizing an antibody that recognizes a cell surface molecule, CD19, present on most lymphomas, B-cell leukemias, but also present on normal B cells. And you can see this first patient who was treated had a complete response, now ongoing 10 years later. You can see these large deposits in the mediastinum that disappeared, these large lymph nodes surrounding the aorta and vena cava, large spleen came down to normal, large iliac nodes which disappeared as well. His marrow was full of tumor and that disappeared, and this is now commonly seen in patients that are treated with anti-CD19. But you pay a price, and this is an important point, because it's just as easy to kill a normal cell as it is to kill a cancer cell. And since normal B cells express the CD19, you can see the disappearance of these B cells within several days after the T cell infusion that stay eliminated for many months before they slowly recover. Now, this is at a time when normal T cells and NK cells are recovering in these few days as the B cells are disappearing. And we've learned this lesson the hard way in many cell therapy protocols, and that is if you target an antigen that's also present on a normal cell, even if you can cause tumor destruction, you will destroy that normal tissue that expresses that antigen at the same time. Well, in the surgery branch, we treated a series of patients and saw objective response rates of 73%, durable complete responses in the 40% range. A former fellow of mine came to see me after we first reported this data in 2010. The patient was treated in 2009. Ari Beldegran came to see me, was interested in seeing whether or not this kind of treatment can be commercialized. Multiple pharmaceutical companies had come to see me earlier and had said, hey, if we have a lymphoma, we'll come see you, but we don't see how we can make money doing something this complicated. But Ari Beldegran had the vision to try to make this commercially available, as did people at Novartis. He performed a multi-institutional study of 101 patients to reproduce these results. In 2012, when he came to see me, we signed a cooperative research and development agreement to transfer our technology to Kite Pharma. They received FDA approval in 2017, and in 2017, in October, Kite was sold to Gilead Sciences for $11.9 billion, who have now made this treatment available worldwide. And Novartis, of course, has it available as well. And so this became the first cell and gene therapy ever approved by the FDA, and there's worldwide interest in this kind of approach. A review put together that I've recently seen showed 200 different companies around the world trying to develop cell therapies. Well, the problem of cancer in the United States is summarized in this slide. About 600,000 deaths a year due to cancer. 90% of those deaths are due to the solid epithelial cancers, the remaining 10%, the hematologic malignancies. It's these solid cancers that start in tissues that have glands, and it's the epithelial lining of these glands that are constantly turning over. As they turn over, DNA replication errors are made, mutations, and it's the accumulation of these mutations that leads to the cancer phenotype. And so the major challenge that confronts oncology today, not only cancer immunotherapy, is the development of effective immunotherapies for patients with the metastatic solid epithelial cancers that cannot be cured by any available treatment. Most of these patients are not susceptible to treatment with the checkpoint modulators and result in about 90% of cancer deaths. Now, I brought up the CAR T cells because there's so much confusion now about the value of CAR T cells for the treatment of solid epithelial cancers. CAR T cells require the use of monoclonal antibodies that recognize molecules on the cell surface unique to cancer. Monoclonal antibodies were first described by Kohler and Milstein 45 years ago, and no monoclonal antibody has been identified that recognizes cell surface molecules unique to cancers and not present on normal cells as well. And again, normal cells expressing this, as I've shown, are highly sensitive to destruction. And so right now, it does not look like CAR T cells are useful for the treatment of these solid epithelial cancers. And unless there are some new innovations, I think we're not likely to see progress in this area in the short term. It's the conventional T cell receptors that we've emphasized to try to treat these common epithelial cancers. And I want to talk about those predominantly, but we've learned such important lessons from patients with melanoma that I want to start by spending a few minutes on that. We utilized TIL cells, tumor-infiltrating lymphocytes, that we would isolate from a cancer deposit and enable them to grow in vitro under the influence of IL-2, and use those bulk TIL cells without any selection for anti-tumor cells to treat patients with metastatic melanoma. And we've widely reported this, but this is an update to the present time, adding about 90 patients to what we've previously reported. We've treated 194 patients with metastatic melanoma with their own tumor-infiltrating lymphocytes grown in vitro and transferred to the patient after the patient receives a lymphodepleting non-myeloablative chemotherapy that eliminates lymphocytes and white cells for about 8 days before they recover. And as you can see, our objective response rates by classic RECIST criteria, 55%, about a quarter are complete regressions. And the hallmark of cell therapy is the durability of complete regressions. And as you can see here, in these 46 patients that had a complete regression, only 2 have ever recurred to the present time, with a median potential follow-up of well over 7 years. I'd make 2 points. One, there's no relationship between the bulk of disease and the likelihood of having a complete response. And second, all but 2 of these 46 complete responders received just a single infusion. 2 patients required 2 cell infusions. And so this is a living treatment. The cells are infused, they circulate, patrol the body until they see their target, where they stop, expand, and we can see expansions even in peripheral blood of 1,000-fold in the first 10 days after the cells are administered. And you can see that in this curve for the complete responders, except for those 2 patients that recurred. We haven't seen a recurrence in that. Somehow, adoptive cell therapy with TIL appears able to eliminate the last melanoma cell. And so very recently, we've begun studies asking what are the phenotypic characteristics of the actual cells that mediate cancer regression in vivo. And to do that, we've used this high-dimensional single-cell transcriptome analysis that one can perform that can analyze up to 10,000 cells and cluster cells based on their transcriptome analysis. And so this data, which is soon to be published in Science, took 16 melanoma patients and did this high dimensional analysis that formed these clusters from melanoma patient infusion samples into 22 individual cultures that several of which seem to separate responders from non-responders. When each of these clusters were analyzed for responders or non-responders based on the genes that were being expressed, it turned out that only one cluster, cluster number 1, separated responders from non-responders. There were no other clusters that separated that, thereby indicating a gene cluster that was associated with tumor regression in the infusion samples. And it turned out that several critical genes, most importantly CD39 and CD69, were negative in responders compared to non-responders. In fact, if one looked at the total cell number that was administered and looked at melanoma-specific survival, there was no difference, p-value of 0.3, based on the total cells given. But at a very high level of statistical likelihood, p0001, it was the number of double negative cells that were highly expressed in cluster 1 compared to double positive cells that correlated with the regression and increased survival of melanoma patients. And so it looked like these CD39 negative, CD69 negative stem-like lymphocytes were the effector cells responsible for melanoma treatment. And we took that observation then back to the mouse in a P-mel model where we isolated P-mel T cells, re-stimulated them, and at day 10 of growth isolated the CD39, 69 negative cells compared to the positive cells. And as you can see in this tumor area curve and survival curve, when we gave 3E5 of the double negative cells or 5E5 double negatives, we saw an improvement in survival, whereas the double positives, even at the higher concentrations, had minimal impact on tumor growth and survival. And so we're right now looking at ways to purify these CD39, 69 positive cells for human treatment. Well, as we demonstrated, melanoma TIL appear able of controlling the last melanoma cell. And our clue to trying to identify what these cells were recognizing came from the observation that we were seeing specific cancer regression in the absence of all off-tumor on-target toxicities. That is, we saw no normal toxicities in these patients. And that led us to explore the role of specific cancer mutations as the targets of TIL. After all, if you wanted to find something that was different from cancer cells, from normal cells, it would be the accumulation of mutations that led to the cancer. And so we began and developed an assay to mine the cancer exome to identify immunogenic cancer mutations. Now, for a mutation to be a cancer antigen, it has to be processed intracellularly into a small peptide, and that peptide has to fit in the groove of one of the patient's surface MHC molecules, and therefore only very rare mutations will be antigenic. And so we developed this blueprint for the identification of immunogenic mutation-reactive T cells in patients with common epithelial cancers, melanoma, and then these epithelial cancers as well. And we do that by isolating genomic DNA, RNA, and do whole exome and transcriptome sequencing to identify the mutations using modern Illumina equipment that can be done in a routine research lab in 8 to 9 days, the bioinformatics another few days, and within 2 weeks, you can identify every mutation that's expressed in the cancer cell. The key to this assay then is to synthesize 25-mer peptides or many genes that encode those peptides with the mutation in the middle of this 25-mer. We then take those mini genes or 25-mer peptides that contain every mutation, put it in a patient's own autologous antigen presenting cell that presents all of the class 1 and class 2 MHC molecules of that patient. We then take TIL that mediated complete tumor regression, incubate it with this antigen presenting cell, and if the TIL recognize antigens present on this antigen presenting cell, they'll secrete interferon gamma, they'll upregulate 4-1BB-OX40, and thereby identify the exact antigen that's being recognized. And so again, the key is to make a 25-mer with the mutation in the middle. It has to contain every peptide that contains that mutation that could be presented on an APC. The mutation can be the first amino acid of the peptide, or the mini-gene, the last amino acid, but it has to be included in here, and we can string them together in what we call tandem mini-genes to use for identifying these antigens. The advantages of this approach is there's no peptide binding to MHC predictions that are needed. Every candidate peptide in MHC locus is included in the screen, and there's no tumor cell line necessary. The conglomeration of all the mutations represents an avatar of the tumor cell that can be used for in vitro assays. Well, we performed this assay on 76 consecutive melanoma patients. They had a median number of mutations of 318. That was 44,000 different mutations that were analyzed. We only screened 13,000 of them because those were the 13,000 expressed genes, and could identify 180 immunogenic epitopes recognized by the patient's T cells that were presented on that patient's autologous cancers. It was 72% of all of the melanoma patients. 180 of these 13,400 mutations were recognized by the patient's T cells, 1.3%, and 92% were recognized by CD8 rather than CD4 cells, which, as you'll see, is quite different from that in the epithelial cancers. But what was most surprising in this analysis is that every one of these 180 immunogenic epitopes recognized by the patient's own T cells were unique, and none were shared between patients. Well, we then did this analysis on 130 patients with metastatic gastrointestinal cancers. They had a median number of mutations of 123. We screened 15,000 individual mutations to identify 210 immunogenic epitopes, antigens. That was 1.3%, very similar to melanoma. But interestingly, half of these T cells that recognize antigens were CD4, recognizing antigens on class 2, and roughly half on CD8. And again, to me, surprising in retrospect, were all neoantigens that we recognized. 209 of them were unique to the individual patient. Only 2 patients shared the same exact KRAS mutation that was restricted by CW0802, a relatively rare class 1 molecule. We then analyzed 43 metastatic breast cancer patients, analyzed 4,700 plus genes, identified 100 immunogenic neoepitopes, so 67% of breast cancer patients contain T cells recognizing their antigens. 2.1% of all of the mutations were immunogenic. Again, the majority this time was CD4s, and in breast cancer patients, every neoantigens were unique. None were shared among patients. And if we now take 195 patients, adding in ovarian cancer, prostate, endometrial cancer, and so on, if you look down these histologies, the number of patients with neoantigen reactivity is about 80%. And that resulted in 363 total neoantigens recognized. Still, only 2 antigens were shared, and these were 2 KRAS antigens that turned out to be immunogenic. So we generally think about melanoma being immunogenic cancer, and it is, but it's probably true of all cancers, because you can identify T cells in almost 80% of all of the common cancers, and we're getting better at defining them by looking at additional antigenic types. It appears as if most epithelial cancers, the major killers, are immunogenic based on this kind of analysis. Well, an advantage of targeting mutations is its applicability to target multiple cancer types. And so this is an immunotherapy not unique to colon cancers or breast cancers, but basically any patient that might have, that has cancer. And since all cancers have mutations, its potential applicability could be wild type if we just could select the cells that recognize these mutations. And that turns out to be a very significant problem. But to illustrate the targeting of mutations in multiple cancer types, I'd like to share some individual patient stories that will illustrate the fact that by targeting mutations, you can potentially target virtually any cancer. The first patient ever to respond to this kind of approach, a patient in which we could identify fragments from their tumor that were very highly expressing T cells with a unique ability to recognize our antigens, was a 45-year-old patient that had a metastatic cholangiocarcinoma bile duct tumor. She had had a right hepatectomy, developed multiple metastatic deposits, not an uncommon story for this kind of tumor, received chemotherapy, progressed, more chemotherapy, progressed. We gave her bulk TIL cells, the same kinds of cells we use in melanoma to treat her cancer, and she did not respond. We then selected a population of T cells uniquely able to recognize her unique antigen that we had identified, an ERB2IP mutation. She had very few mutations, only 26, but one of them was immunogenic. And she underwent a complete regression of her disease. As you can see, these lung metastatic deposits, three liver deposits were eliminated, leaving a hole, one of them leaving a hole in the liver. And this patient is now disease-free and living normally 7 years later. This patient had metastatic breast cancer that we reported about 1.5 years ago. She had, again, she had hormone-positive cancer and a fairly typical story, moving from one chemotherapy, hormonal therapy, targeted tyrosine kinase inhibitors, progressed through them until she came to the NCI to receive her own T cells that recognized her cancer mutations. She's had a complete response and is now living disease-free almost 5 years later. We recognized 4 random somatic mutations in this patient. They had no particular driver functions, but it appeared to be the accumulation of these, at least immunogenic mutations and others, that led to her breast cancer. And you see here the complete response, this lesion beginning to grow through the chest wall, multiple liver metastases that disappeared completely, again in an ongoing complete response now 58 months later. This patient with metastatic cervical cancer presented with a fungating cervical mass and multiple lung and intraperitoneal metastases. She required radiation therapy, multiple chemotherapy regimens, came to us finally in March of 2013 when we were beginning these studies. It was these early patients that stimulated us to pursue this vigorously. She received 75 billion of her own tumor-infiltrating lymphocytes and underwent a complete ongoing regression, now disease-free 7 years later. And you can see these lymph nodes here in the left panel, indicated by the yellow arrows. This node was occluding her ureter. That's a ureteral stent. She had a complete disappearance. The stent was of her cancer. The stent was removed, and again, living normally now. Finally, a patient with colon cancer that taught us a great deal. A 49-year-old woman who had an aggressive colon cancer, was invading into her bladder, required a bladder partial cystectomy at the time of her colectomy, received radiation therapy and chemotherapy. We resected 2 lung metastases for TIL. She had 9 lung metastases we were following. We used this TMG approach to target our unique cancer mutations. She had 61 somatic mutations. It turned out that her T cells recognized her KRAS G12D mutation. Again, the administration of these led to the regression of 6 of her 7 remaining lung metastatic deposits. can see those here. But one tumor not only did not disappear, but in fact began to grow and resected that tumor. An analysis of that tumor showed that she had lost a segment of chromosome 6, and chromosome 6 was encoding her CW0802 class 1 MHC molecule that presented this KRAS mutation. And therefore, of course, this tumor escaped, teaching us about at least one of several kinds of escape mechanisms that could occur. But she has not gone on to recur and remains free of disease over 5 years later. So we have a long way to go in this, and I've shown you examples of what's possible. When you give bulk TIL in the metastatic solid epithelial cancers, you see no responses. If you can select TIL, that is, be fortunate enough to identify fragment cultures that have a high incidence of the appropriate cell, we did see 3 out of 25 or 12% responses. When we analyzed the cells that were in the patient that we had transferred, we could demonstrate that these cells had gained expression of PD-1 in a large percentage of cells. And if we then did a trial, an ongoing trial now, of 26 patients who received these selected TIL plus pembrolizumab, our response rates are now 23%. All of the patients that we treat have advanced chemo-refractory metastatic epithelial cancers. But one of our major challenges now, and we'll come back to this in a moment, is how can we better select cells that have anti-tumor activity? Well, there are 2 hypotheses that one might consider from some of the data that I've mentioned. The first is that it's the recognition of random somatic mutations, or perhaps some shared mutations like KRAS and P53, that explain cancer regression from most immunotherapies for solid cancers. Interleukin-2, the checkpoint modulators, anti-CTLA-4, Ton Schumacher, showed that a good responder anti-CTLA-4 recognized a unique mutation. Of course, with the checkpoint modulator PD-1, there's a close correlation between the number of mutations and the likelihood of responding, and certainly these tumor infiltrating lymphocytes. The second is that I think we finally understand what a cancer antigen is. It's any intracellular protein can potentially be a cancer antigen if it's mutated and processed intracellularly to a peptide that can bind to the patient's autologous MHC molecule. And it turns out that only about 1 in every 70 mutated neoepitopes are neoantigens, but they can be identified using appropriate assays. So this is good news and bad news. The good news is since all cancer patients have mutations, they're potentially eligible. But the bad news is that the treatment is going to have to be highly individualized for each patient and thus complex. And I often hear that as a criticism of this kind of approach, but I heard it through when we began our studies with CAR T cells until somebody came along and developed simplified techniques for developing the CAR T cells. And I think as we learn more, this kind of approach can be more widely applied outside of sophisticated research laboratories by sending tumor deposits to potentially to a central facility where the kinds of assays that I mentioned can be used to identify the T cells that recognize these tumor antigens or the T cell receptors that can then be shipped back to a home institution. And there are many commercial companies that are trying to identify this kind of treatment approach. Well, I've talked about the unique somatic mutations, and we've talked about non-mutated proteins on the surface of cells, such as CD19, but again, there are very few candidates for the solid epithelial cancers that are unique to the cancer. But there are driver oncogenes, or tumor suppressor genes, that are shared among the patients. And we've only found one responding patient to KRAS. And it is surprising in the field of oncology how few shared antigens there are. There's KRAS and P53, by far the two most common. In melanoma, there's BRAF, about half of melanomas. But when you get to other shared antigens, you're talking about low single digits. And so we've tried to see if we could target the large number of patients that have KRAS and P53 mutations by identifying T cell receptors that can recognize these mutations. KRAS is a GTBase protein that's essential for normal signaling. Activating mutations are essential for many development of many cancer types, and there fortunately are hotspots at the 12th, 13th, and 61st position of the KRAS molecule that account for greater than 80% of all KRAS mutations. And so we've developed approaches to try to screen all KRAS mutations that are G12D, V, or G12R to see if we could identify T cells that actually recognize those mutations, either naturally or by further in vitro sensitization. And you can see in this study that in fact we've now developed a library of KRAS mutations that recognize KRAS on the basis of presentation on class 1 or class 2 molecules. Now, of course, even though there are hotspot mutations, because they're presented on different MHC molecules, you need a T cell receptor that's unique to each one. And so we're starting to develop libraries of these T cell receptors. In our current library, if we can use those T cell receptors transduced into a patient's lymphocytes, potentially recognize up to 29% of patients who have KRAS mutations present in their cancer. We've also concentrated on P53, a tumor suppressor gene that's present in 50% of human cancers. It's the most frequently mutated gene. And although it occurs throughout the DNA binding site of the P53 mutation, there are up to 10 major hotspots that account for a significant percentage of mutations in these patients. And so we've developed a library of T cell receptors that recognize a variety of different P53 mutations, largely hotspot mutations, but some non-hotspot mutations that can also potentially be used as an off-the-shelf T cell receptor to treat patients in ways that might be similar to that of CAR T cells. None of this information has been published yet, but we're preparing it for publication to make all of these sequences available to the community at large, and certainly can send them to anybody that is interested now. This would account for almost 6% of all patients with P53 mutations. And so I'd like to tell you about the first patient in which we took this P53 T cell receptor present in a patient's cell to attempt to use it to treat a breast cancer patient. This was a 48-year-old female who had breast cancer, who had had a surgery, radiation therapy, a whole variety of chemotherapy agents and hormonal treatments. She came to us with extensive disease. In fact, when she actually presented to us for treatment after we had decided that we could potentially target her P53 mutation, she had a cardiac tamponade. Her pericardium was completely replaced by tumor. She needed a pericardial window placed, which enabled us to biopsy and prove that, in fact, it was all due to this P53 containing cancer. We treated her with 53 billion of our own T cells, and she had a dramatic partial response, but did recur at 9 months, and we're now in the process of trying to analyze the escape mechanisms that occur in these patients. But she had a dramatic response. You can see the surface of her breast and extending into the opposite breast, replaced by cutaneous and subcutaneous tumor, with tumor on the chest wall as well, all of which went away for 6 months. She had a pericardium full of cancer that reverted to normal for 6 months, and of course her pericardial effusion went away as well. And you can see in this patient, breast almost replaced by tumor nodules extending onto the opposite breast that disappeared for about 6 months. So we're attempting to now, and this is again very work literally ongoing as we meet together. We try to use these high-dimensional single-cell transcriptome analyses to identify gene signatures that might indicate the T cells that are capable of recognizing a cancer. And so we did that kind of analysis in a patient with rectal cancer, looking at the gene clustering. Because we had identified the antigens and the T cell receptor sequences from this rectal cancer patient, we could then overlay the T cell receptor sequences, and it turned out they all fell into this one individual cluster. Breast cancer patient, again, we knew the T cell receptors and could now backgate onto this particular cluster. Apparently, the overwhelming majority of the receptors did fall in that one cluster. And then when we did this analysis for 9 cancer types, rectal cancer, breast cancer, some melanoma patients, you could see again, and when we backgated the T cell receptors back on to these clusters, we could identify these clusters that contained the T cell receptors that recognized the cancer antigens. Well, we could then look at those clusters for neo-TCR signatures and project those signatures based onto clusters. And in fact, by isolating the T cell receptors in those clusters that were not previously identified, we could identify new T cell receptors, and did that for a couple of tumors, and could identify genes in a neo-TCR gene signature led by CXCL13, as well as other interesting genes that could identify T cells that did contain T cell receptors capable of recognizing antigens, and we're studying ways to try to isolate them. Well, what I've tried to present in this brief discussion is a blueprint for cancer immunotherapy directed against the common epithelial cancers by targeting the immunogenic somatic mutations unique to the autologous patient's cancer, and by raising libraries of T cell receptors against shared cancer mutations such as KRAS and P53. And so I would conclude with 3 tentative conclusions. 1, that cell transfer therapy can mediate durable regressions in patients with metastatic cancer refractory to other treatments. The T cells that recognize unique somatic mutations can be found in TIL, and we've demonstrated and published that they can be found in peripheral blood as well. Interestingly, about 1 in every 1,000 TIL can recognize antigens, but 1 in every 100,000 PBL will recognize those antigens. And finally, that the identification and targeting of mutations unique to each cancer, or shared mutations such as KRAS or P53, have the potential to extend cell therapy to patients with common epithelial cancers. Well, thank you for your very kind attention.
Well, thank you, Dr. Rosenberg, for a really exciting talk. And it's just extraordinary to see those responses that you have in patients and how durable they are. It's incredibly impressive to me. We have some questions now from the audience, but please, for those of you who are with us, please use the Q&A feature to enter questions for Dr. Rosenberg. I'll start reading one. Someone asked, could Dr. Rosenberg comment on non-viral gene editing delivery approaches for TCR TILs, such as CRISPR and transposons?
So we've used CRISPR to knock out genes. We're looking to identify internal checkpoint modulators and have concentrated on the cis molecule. It's a member of the suppressor of cytokine signaling family, and when you knock it out with CRISPR, you can improve treatment in mouse models, as published by Doug Palmer, a fellow in the surgery branch. But we haven't used CRISPR to insert T-cell receptors. We are, however, working intensively on trying to use Sleeping Beauty transposon transposase systems to introduce T-cell receptors, and have in fact worked out methods to do this with high efficiency. We're now trying to polish that approach to insert T-cell receptors. We're developing clinical protocols actually to actually use these non-viral genes, which would simplify the kinds of procedures necessary to do this without having to produce GMP quality viruses. And so I think that's an approach that has some potential application and some promise as well. Right now,
Great.
Everything we're doing is with retroviral gene insertion.
Thank you. Another attendee asked, the Kite and Novartis CAR-T products are extremely costly for patients, several $100,000. Do you anticipate that costs can be reduced with autologous products to make it available to more patients?
These are very expensive treatments, no question about it, but it appears that they can be curative. And the best way to save healthcare dollars is by curing patients and not having them go from one ineffective treatment to another that turns out to be a lot more expensive than the few $100,000 of CAR T cells. Now, of course, I have no involvement with these individual companies, but what we're seeing now is the first manifestation of the application of cell therapy. There are multiple companies now that are trying to develop off-the-shelf T cells, allogeneic T cells, into which CD19 CAR receptors can be introduced by eliminating class 1 molecules and preventing graft versus host disease by eliminating the endogenous T cell receptor. So I think there are ways that it can be simplified. And in fact, it takes us about 28 days to produce those cells, or the cells used to treat patients with epithelial cancers. We've now reduced that time down to 12 days, which reduces a lot of the reagents that are necessary. So I think as the genius of American industry gets involved in trying to develop treatments that they can commercialize, we will see a simplification and we will see a reduction in cost.
Great. That covered the next question, which was asking about allogeneic CAR-T. What are your thoughts about targeting aberrant splice variants as a possibly larger source of neoantigens, especially in those 20% of patients with no TILs expanding in the TMG assay?
There are a variety of other chromosomal abnormalities that, other than just a random somatic mutation, that might give rise to immunogenic phenotypes. And Vid Leko, a postdoctoral fellow in the laboratory, and I have just, in this issue of Cancer Cell, published a review of a variety of cancer antigen types resulting from splice variants and other sources of antigens. One that we're very interested in now are mutations in mitochondrial DNA that can be expressed in the cell and potentially be immunogenic. That's certainly been reported by others, and we're pursuing that now. So I think there are other kinds of antigens and other chromosomal aberrations that can give rise to these, and they need to be explored as well.
Great, thank you. Dr. Crystal Mackel is saying, thank you, Steve, for sharing your exciting work today. Given that most of the immunogenic peptides are derived from passenger rather than driver mutations, there would be expected to be a risk for antigen escape. Have you observed this in non-responders or patients who progress after response? What do you know about resistance in these studies?
So a lot of our efforts now are trying to define resistance, but Dr. Mackel raises a very important point. Because as tumors metastasize, they change, and one ends up with different clones within comparing different metastatic deposits, and even within an individual tumor resection, you can look at different parts of that cancer and find different antigenic variants. And so That's a problem that we're trying to overcome several ways. One, everything we've done up to the present time, we utilize a resected metastatic deposit to identify the mutations. You can do this complete mutational and whole exome analysis on fixed paraffin tissue. And so we're going back to the original resected tumor that presumably does contain the driver mutations, which should be expressed or commonly expressed in metastatic deposits, to expand the number of variants that we look at. We've also seen a significant correlation between the number of antigenic variants that are targeted in melanoma and the likelihood of having a tumor response. And so it's by targeting multiple variants at the same time, multiple mutations, that one might be able to overcome resistance in much the same way we use combination chemotherapy to treat patients. But Dr. Mackel has identified a problem and one that we're vigorously trying to pursue as well.
Great. Thank you. Ron Levy's asking, Steve, do you have an approach to address MHC loss as a mechanism of escape from the TCR-directed therapies?
We don't. Ron Levy, I should mention, was my first laboratory mate when I came to the NCI during my surgical residency. So Ron, it's good to hear from you. No, if the tumor loses MHC, then we cannot target them with conventional T-cell receptors. But there are a variety of investigators and commercial companies that are trying to look at natural killer cell killing, gamma delta cell killing. And so although we've had no experience with those approaches at all, they certainly are being pursued elsewhere.
Ravi Majetti is asking a follow-up question about your treatments targeting passenger mutations, and he's wondering if a polyclonal approach is potentially going to be more successful than targeting individual mutations.
Well, by polyclonal approach, my guess is he's talking about targeting multiple mutations at the same time, which is one reason why I presented that breast cancer patient. And that is, it appeared as if the major antigens that were being targeted were 4 relatively random somatic mutations that did not have any driver or oncogenic potential. And so I think that polytargeting, that polyclonal approach using T cells or T cell receptors that can target multiple antigens at the same time might help overcome the kinds of antigenic heterogeneity that represents a resistance mechanism.
Thank you. Nathaniel Gray is asking if you've looked for neoantigens formed after treatment with covalent inhibitors, targeted therapeutics like EGFR or BTK inhibitors, or the covalently modified peptides.
We have not, but interestingly, if you look at the T cells that were in a resected tumor and identify antigens that are recognized, if you then administer those T cells to a patient and follow their persistence, and many persist for many months or years, you actually can then identify T cell receptors that recognize antigens that you didn't recognize in the original, from the original tumor. And so there may be some kind of antigen spreading that takes place following this therapy, or maybe other therapies. And in fact, one of our fellows in the lab now is looking to see whether or not there are individual chemotherapy regimens that are considered antigenic friendly. I don't know exactly what that means, but it's a term that's been used to see if in fact there are certain kinds of therapies like individual chemotherapy agents, radiofrequency ablation, stereotactic radiation therapy to individual lesions at high doses, to see if they can in fact broaden the antigenic repertoire presented by the cancer deposit.
Thank you. Amin Alipur is saying, thanks, Dr. Rosenberg, for the discussion. Any thoughts on how tumor delivery or infiltration could bottleneck these approaches? Some in the solid CAR T field have resorted to intratumoral injection only.
Well, intratumoral injection can be very powerful and is pioneered in many ways by Ron Levy at your institution. With respect to T-cell administration to localized tumors, in general, if the localized tumor is the only problem, you can take it out. But it's a metastatic disease that has to be targeted, and there are approaches, and I've mentioned Ron Levy's work, that look at the ability of these intratumoral injections to extend the therapeutic potential to other tumors. But we ourselves have not looked at intratumoral therapies. The patients that we treat have widespread disease.
Thank you. Stephen Feldman is asking, have you demonstrated that the CD39 negative, CD69 negative T cells contain the majority of tumor reactive cells in the infusion bag? And what is the percentage of these cells in the bag, and can they be developed for a TIL therapy?
So it's good to hear Steve Feldman, who developed a lot of our T-cell receptor work here at the surgery branch before he went to Stanford. So Steve, it's good to hear from you. These CD39 negative, CD69 negative cells are a very tiny percentage of the population. Most of the effector cells in the infused cell product are CD39 positive, CD69 positive, whereas it's the double negative cells that correlate with the therapeutic effect. And they appear to be a very tiny percentage of these stem-like cells that have an extraordinary proliferative potential when administered to the patient, where they turn into the CD39 positive, CD69 positive cells that mediate the tumor destruction. And we're now, again, that information is very new. We're now trying to develop techniques to isolate CD39, CD69 positive cells, as we did in the mouse model that I mentioned, but to do that in the human and generate enough cells that we can use for therapy without having them differentiate into positive cells before they're administered.
Okay, thank you. David Miklos has an interesting question. He asks, why do you think Rick Child's efforts at NCI in 2000 using allogeneic hematopoietic cell transplant to treat renal cell carcinoma and melanoma failed to provide benefit? Do you think that allogeneic transplant may have developed neoantigen responses?
Well, actually, Rick Childs reported that in a small percentage of patients, he did see responses when doing allo transplants. And the mechanism is not known, but if you want to stimulate a lot of cytokine secretion in a patient, do a transplant. And interleukin-2 administered alone can stimulate anti-tumor T cells to mediate tumor regression in about 15% of patients with melanoma and renal cancer. And so that might be the mechanism, and that is the transplant itself stimulating enough cytokines that other cells, not the transplanted cells, but other endogenous cells could mediate a tumor response. But Rick Childs and I disagree a little bit on the possible mechanism, so you'd have to address that question to him.
Thank you. Sunil Reddy is asking if you can comment on brain metastasis and immunotherapy, and asks what sort of brain metastasis patients can be treated effectively with your treatments.
So we've used our cell transfer therapies in patients with brain metastatic deposits. And the response of tumors in the brain is lower, about half of that in other visceral sites. And we published several papers. Rick Sherry from our group has presented that kind of data, but they're clearly less responsive and have a much higher tendency to recur at other sites when these brain metastases do recur. So that represents a potential problem. For our epithelial cancer patients, because of the increased toxicity of the lymphodepletion, which also can affect platelets in patients with brain metastases, as we're developing this in epithelial cancers, we're not accepting patients that have brain metastases as part of our eligibility criteria. So the eligibility criteria for those epithelial cancer patients are less than 70 years of age, fairly reasonable performance status, and no brain metastases.
Thank you. Rob Negrin is asking, do you see a strategy to develop a synthetic T cell to avoid extensive culture experiments that are expensive and patient-specific?
Well, I guess it would be possible. The closest approach to that today is the use of allogeneic T cells. And so, for example, commercial companies are pheresing young, healthy adults. From a single pheresis, they can then, by genetic manipulation, eliminating endogenous T cell receptors, class 1 and class 2 antigens, they can develop enough off-the-shelf modified T cells into which they put anti-CD19 CAR to treat 100 patients from a single pheresis of a young healthy donor. So in some sense, that's a synthetic cell. And a variety of commercial companies are trying to look at that approach, which they believe can cut the cost by a factor of 3 or 4.
Okay, well, we're at the top of the hour. So thank you, Dr. Rosenberg. It was wonderful hearing your exciting science, and thank you for joining us. I wish that you could have been here in person with us for more intimate interactions, but it was wonderful meeting you, and thanks for joining us at the Stanford Cancer Institute.
Well, you're very welcome. I look forward to visiting you soon.
Okay. And then brief announcement, our next Frontiers in Oncology is going to be on November 17th. Dr. Robert Weymouth, the Robert Eccles Swain Professor of Chemistry at Stanford, will be presenting. Thank you all and have a great day.